Okay, our next speaker couldn't make it here, Dr. Ullas from TIFR. He's being represented by Shreyam. So Shreyam. So I'll probably stand up after 10 minutes so that we can wind down by 12 minutes and then have more questions at the end, okay?
Yeah. Hi everyone, good afternoon. I am Shreyam. I am a master's student in Professor Ullas Koltur's lab in TIFR Mumbai. So today I will be talking about metabolic sensing and plasticity. So before I move on to the main presentation, I'll just give a head start to what I mean by metabolic sensing and plasticity per se. So metabolic sensing basically refers to sensing the status of a cell, whether you are energetically starved, whether you have plenty of energy, whether you are stressed, unstressed. In that context, I will be talking about metabolic sensing. And what I mean by plasticity is that whether you are able to transition from one state to another. For example, if you are exercising, you want to quickly transition from a rested state to an active metabolizing state. Or if a stem cell starts proliferating, you again have to take a quick decision between transitioning from a quiescent state to a proliferating state. So that's what I will be meaning by metabolic sensing and plasticity. Now, space travel, as we were discussing till now, it's obviously quite stressful for the body. First of all, there is microgravity, radiation, physiological issues because of isolation. Now, coming to the specific problems which astronauts face while in space, firstly is mitochondrial dysregulation, which I will be definitely talking about in details in the further slides, so I'll skip that for now. Second is oxidative stress. Due to radiation as well as excess metabolic flux, there is always a ROS generation which is there in astronauts when present in space, which can severely affect the whole body physiology. Then we have DNA damage, which is again very obvious. So astronauts generally face radiations which are multiple times higher compared to that present as we are on Earth, even after the protective gear which they use. Even after that, they do experience DNA damage. Telomere shortening, which again I won't be much going into. There is muscle atrophy, which is a rampant problem for all astronauts, which is why they exercise before a flight, after they come back from flight. Compromised immune response, as we discussed in previous presentations. Metabolic defects, obviously there are lipotoxic pathways which have been shown to be activated when in space. And finally, endocrine defects, insulin and diabetogenic phenotypes often come up in astronauts after they come back from space. Now, as I said, I will be focusing mainly on mitochondria. So there is an intricate interplay between diet, mitochondria and microgravity going on here. If you see here, this paper in Cell which came out in 2020, by multiple omics analysis, they showed that astronauts after coming back from space, there are increase in ROS levels, glycolytic TCF flux, whereas there is a decrease in the cellular energetic status, including the ATP as well as the proteins involved in the electron transport chain, which actually generates the energy. Now, what we as a lab we have been studying for a decade now, is basically this. We have shown that under multiple paradigms, including diet and dietary restriction and calorie restriction, there are effector proteins which sense the status of the cell. For example, here I have just put 31, and these proteins are known to actually ameliorate these exact defects, not under the influence of microgravity. That has not been shown, but the same phenotypes are also mediated by these effector proteins themselves. So, and thereby it mediates aging and metabolic plasticity. Now, the interplay, what we and multiple other peoples have also shown is that this interplay leading to this plasticity and memory is responsible for sustenance of phenotypes. For example, what I mean here is that if these transitions in plasticity are hampered, you will often develop phenotypes such as diabetes, which is a rampant problem. And as I said, as I mentioned, showed in previous slide, that these are effects which have been shown to be rampant in astronauts after they come back from space. And yeah, so that is what we majorly study using multiple across evolutionary cohorts, including humans, mice, fruit flies. We basically study the interplay between diet physiology and metabolic and physiological plasticity. Now coming back to microgravity itself, there are a few very important open questions here. First is, what are the long-term consequences for spaceflight on metabolic and physiological fitness? By long-term, what I mean is is throughout lifespan. Now fruit flies become a very nice model for assessing the long-term consequences, because fruit flies have a very short lifespan, which is about 90 days. So we send them to space, they come back, and we can span their entire lifespan to study exactly what is happening and whether there is a memory associated with the early spaceflight exposure. Second, the impact of shorter duration space travel on whole body physiology. So the Gaganyaan mission, as we previously said, it can span any time from 1 day to 7 days. Now in fruit fly model systems, mainly what has been studied is around 14 to 15 days of space travel. So again, the Gaganyaan mission does give us a very useful model to study shorter duration space travels, which will become very relevant because of the current scenario in which people are starting to go back to space for short travels in low orbits. And third is, as I discussed this, how does the memory of spaceflight impact physiological plasticity upon return? Now, memory of the memory of spaceflight has been studied. I won't say it has not been studied, but there are issues with that. Mostly there are inconsistent data due to the lack of statistically relevant studies and very small sample sizes. So you send flies, they come back, and many of them are dead, which is why the sample size becomes too small for you to do a statistically relevant study, which is why more research on that front is obviously required. Now what we propose here is that on a short-term objective, one of our short-term objective is to use a genetic perturbation model, which is a SIRT1 overexpression. So SIRT1, as I mentioned previously, it's just another metabolic sensor. We need not know the details of it, but it is known to ameliorate the ill effects, which is similar to that observed in microgravity, and there are also multiple pharmacological agents which are known to be inducing activating SIRT1, as well as dietary perturbation, very established dietary perturbation protocols which are known to activate the SIRT1, which is one of our key aspects of studies. What we also want to understand is the fundamental biology which is affecting which is causing these phenotypes. So we know that multiple of these things happens, but how does it happen is an important question. What are the main signaling cascades, the nodal points, which is deregulated due to the memory of this microgravity? Finally, I have the experimental design here. The flies will be sent to space in the same module as discussed by Dr. Ravi. After coming back, what we plan to do are next generation sequencing studies, metabolomic studies, physiological assays throughout their lifespan. So it's not just they come back and we do the study. They come back, we let them age and then do the study to see whether there are any long-term consequences to the memory of this space flight and if the plasticity is lost or not. In future flights post this Gaganyaan, we also plan to send mouse models, obviously there will be, and we want to study light-dark cycle effect of diet, exercise conditions, where we know we already have the expertise of doing respiratory exchange ratios, where we study the circadian cycles. That is one of our future perspectives. Finally, coming to the acknowledgement, I would like to acknowledge Professor Ullas for giving me the project, and the UK lab members past and present, DBS TIFR, scientific kitchen staff, and ISRO for funding. Thank you.
Thank you, Shreyam. It was well ahead of time. So we have time for some questions from the audience.
Hi. So you said once the flies come back, you will let them age and then study their stress levels. How will you determine whether the flies that you're detecting after aging is normal, where initially affected is only or not? Or like?
Yeah, so we have ground control studies. These are studies which we have already done and published, where we already have shown that an age of around 35 to 37 days is a old enough age to induce physiological changes in flies, where they show reduced negative geotaxis ability, and their plasticity to transition from fed to fast to refed states become affected. So the age we have already determined by ground control studies in previous published papers from our lab.
But from that you get the number of days, right? Exactly. But when you're looking at a particular fly, for instance, how do you know it was originally affected in the first place?
Originally affected by
the
microgravity.
space, right? Yeah.
So that is an interesting question here. There is no way we can say, once we are sending, we know that they are in microgravity. Now, if they do not show any phenotype, the obvious interpretation would be to say that it is not being affected, but then again, we would have to repeat it in multiple experiments to finally say that it is not affecting. But there is no way to specifically say that if they're going to microgravity and coming back, they are being affected. There are published data sets on NGS showing that specific pathways are being deregulated, but they are not repeated enough number of times for us to confidently say that this is one of the major nodes which is always being affected once you're in microgravity.
Okay.
I think we had a question from. Yeah,
Hello.
go ahead.
I had a question about the control for this. So, would you be having a ground control where you will be feeding the same diet that you give to the flies on flight?
Yes. So, previous studies from our lab, we have a very specified diet protocol which we give to flies for our optimum lifespan, and there will be ground controls which will be fed the same diet for this as the flies in space.
Okay. Also, one more question. Since they are in microgravity conditions, would that be highly stressful for the flies while feeding and all? Would they always be like floating in the air or having to fly?
So, previous papers have shown that microgravity exposure does affect feeding behavior where flies tend to feed less. So, obviously that effect will be there, but exactly that is what we are trying to study. That is the biology of space travel. If you go, there is a phenotype of less feeding, then after you come back, even if your feeding cycles become normal again, are you still able to maintain a normal physiological homeostasis as the ground controls?
Okay. Yeah. Thank you.
Okay, one last question.
So just to clarify, Sirtuin-2, right?
Sirtuin-1.
Sirtuin-1. So this is not like Sirtuin-2, which is involved in heterochromatin silencing.
Sirtuin-1 is also involved in heterochromatin silencing, but it is also involved in multiple other things.
And you're overexpressing it.
Yes.
So by any chance, does that affect in increasing longevity?
Yes, it does. Sirtuin-1 is known to be a longevity promoter. So yeah, it definitely does. So that is the idea behind it. We are hypothesizing that once they come back, the control flies, we will obviously send an overexpression set and a control set. The control set, we are hypothesizing that it will show a reduction in lifespan. And if whether or not Sirtuin-1 is being able to ameliorate it or not.
I see. Thank you.
Okay, so thank you very much. Please thank the speaker again.